So the topic is a more sustainable phosphate fertilizer. And so I'm going to begin with my definition of this. It is up there, wonderful. So as you can see, the definition I'm using is sustainable phosphorus means optimizing P use so that abundant, nutritious food coexists with healthy aquatic ecosystems. Now I'm going to come back to this definition in just a couple minutes as I launch into the technical part of my talk. But before I do that, I do want to introduce to you an organization called the Sustainable Phosphorus Alliance, or SPA, S-P-A. This is a relatively recent organization that was founded by Arizona State University to promote sustainable phosphorus. And it's a member-funded, multi-stakeholder organization to help the whole world make its use of phosphorus much more sustainable. Down at the bottom of the slide, you'll see the founding members of this. We have additional members that are not on the slide, but these are the founding members, and I'm very happy to see that OCP is one of them. So what do? We try to help people do a better job of making sustainable phosphorus in the whole process from mining to use to recovery, all of it on this. For example, we hold annual conferences. We have one coming up next week here in Washington, D.C. I know a few people in the room besides me will be at this Phosphorus Forum 2017 on it. So that's one of our focus events. In addition, we try to do things that help people who are interested in making phosphorus use more sustainable. We have workshops, and also if you're interested in not only doing more sustainable phosphorus, but being recognized for doing more sustainable phosphorus, then SPA is a very good organization to help with your branding. So that if you're interested in SPA, please contact me either today at the conference or by email later, and I'll get you in contact so you can join us and we can join you. Okay, now back to the definition of sustainable phosphorus and into the technical aspects of my talk. So sustainable phosphorus means optimizing P use so that abundant, nutritious food coexists with healthy aquatic ecosystems. You can see where I put the emphasis at this point. So healthy aquatic ecosystems is the driving force behind why we really need to make phosphorus in all aspects more sustainable. So here is a picture from a very, very famous study done in the 1960s by Dave Schindler. His picture is there. He's a professor at the University of Alberta in Canada, and he did a study in which he divided a lake in Canada into two parts, and you can see the dam dividing it. On one side, he added carbon and nitrogen, and the other side, he added carbon, nitrogen, and phosphorus. And you can see one is really green. That's the one where phosphorus is added. So this was just a groundbreaking study documenting the important role of phosphorus in spurring excessive growth of microalgae or eutrophication on this. Now, around the world, at least in the United States and Canada, we did many, many things to remove phosphate from detergents, to treat wastewater, and we thought we really had things pretty well solved for many of our sensitive lakes. However, at the beginning of this decade, things changed. And so here is a photograph, an aerial photograph of Lake Erie, which once again has very, very serious problems of eutrophication, and these are caused by the runoff of phosphorus from agricultural fields. So the problem has become unsolved, and we know why it's unsolved. Now, this isn't just a problem of Lake Erie or a few places around the world. This is a widespread international water quality challenge. And so here's a heat map showing the anoxic zone in the Gulf of Mexico near the outlet of the Mississippi River in the southern United States. But this is only one. There are more than 400 of these hypoxic, almost dead zones around the world. So this is a giant international problem. And it's one that is caused primarily by the runoff of phosphorus from various agricultural activities. Now, a key thing to keep in mind is that we mine a lot of phosphorus. Most of it goes into fertilizer, and most of that phosphorus does not go into our food. So Donna Cordell and her colleagues in Australia did some fabulous work on doing a mass balances on phosphorus globally, and in 2009 they published a landmark paper that I show you here, in which they showed that only about 16% of the mined phosphorus globally ends up in the food. The rest, most of the rest, ends up either in agricultural runoff from the fields or in animal waste. And it's this lost phosphorus, the stuff that never makes it to our field, that's getting into the waters, causing the eutrophication, hypoxia, and dead zones. We did a paper in 2011 in which we adapted the work from Cordell and produced this table here, and it just shows you in the red where are the big places where the phosphorus is getting out of the human and agricultural system and into the waterways, causing these problems. And you can see 46% from soil erosion and runoff, 40% from animal waste, and about 8% from human wastewater. So most of it is coming from the food sector directly. Now, I'm going to show this diagram a couple times in my talk today. And on the left-hand side of this diagram, you will see three characteristic sources of phosphorus that get out in the environment. The top one is what we call high phosphorus, high BOD concentration. These are high refers to the concentration in the stream. And this, the classic example of this would be animal waste. Very high in phosphorus, very high in organic material. The medium one would be things like human domestic wastewater. And then the bottom one is low phosphorus and low BOD concentrations, and this would be characteristic of your agricultural runoff and drainage. Now, I'm going to start off very briefly in talking about the high and medium concentration ones. And this is not the main point of my talk today, but it provides really important context when we look at what are we going to do to make phosphorus more sustainable. So because these have a lot of BOD or organic material, these waste streams also contain a lot of energy. And so we can convert the organic material into valuable energy forms, as I show up at the top right, things like methane or hydrogen, and at that same time releases organic phosphorus into becoming inorganic phosphorus or phosphate that is recoverable. And so this is a really great system here because we get at least two major benefits out of the material, energy and phosphorus. Well, in fact, we get more than that, as I'll show you. But the key thing I want to point out today is there's a very important take-home lesson for these high and medium strength waste streams is that we now today in the middle of the 2010s decade, we now have technologies that allow us to treat these anaerobically to capture the energy value either as methane through methanogenesis, a very long-standing process, but that has been greatly improved in the last few years so that now it works well for all of the waste streams that were in that top group. Or we can use some more emerging technologies in microbial electrochemistry, that's the second bullet there, where we can generate electrical power or hydrogen gas or hydrogen peroxide or some other materials that have higher economic value than does methane. Now these ones in the second category, the second bullet, are not ready for commercial application yet. I mean, these are still emerging research and development technologies, but they have a lot of downstream potential, and our group and many others are working on it. Again, when you convert the organic material into energy, any of these energy forms, you release the phosphorus as inorganic phosphorus, and it can be captured by a range of techniques that are available now. They're all available now. None of them have been fully optimized for this application of recovering phosphorus, but we have the technologies out there that are being readied for this great application. So if you look at the economic question here, economic balance on these, you ask, well, is it worth our effort, our economic effort to do this? And so our team has done some detailed techno-economic analyses of these systems where we take these wastewaters and we capture the energy, we capture the phosphorus, also the nitrogen and the water. And so this diagram here is a bar chart showing the costs and the benefits of doing this. So the left two bars here, the left two bars are using methanogenesis to capture the energy value as methane gas, and the left bar are all the benefits from the energy, the benefits from the recovering the ammonium and the phosphate, as well as recovering the water. The right-hand bar are all the costs, the capital costs and the operating costs. And what you can see is that the operating costs are less than the benefits. So this is actually a moneymaker, and this is for a particular scenario of about 6,500 dairy cows, and this would be making an operation like this about $200,000 per year. If we could develop and commercialize the technologies, the microbial electrochemical technologies to make hydrogen gas, then we move to the right-hand bars. We get a much, much higher economic value out of the energy, and I'll make almost $1 million per year on that. Now, the take-home lessons here on this is that, one, energy is the main economic driver in either setting. The biggest economic value comes from the energy. However, the nutrients are significant. And in the left-hand two bars, it's actually the amount that turns it from being cost neutral to actually cost positive. Okay, so that's all I'm going to say about these high and medium strength ones. What I'm really going to talk most about today is the bottom one, and that is low phosphorus and low BOD, the runoff and drainage. This doesn't have organic material in it. It's not going to generate any energy, and so you can't do that. So we're basically relying on the water and on the nutrients to provide economic value. So again, no energy output. That's a key take-home lesson. You're not going to get the energy value from it. There is none. Now, my conclusion, when I think about this bottom category, is that recovering phosphorus from the runoff is difficult. It's actually relatively easy to recover the phosphorus from the animal waste and even domestic sewage. It's hard to do it from the runoff for three reasons. One, I've already told you, there's no energy. We can't gain an economic benefit from capturing the energy because there is none. Second, it has a relatively low P concentration, so that makes it technically more difficult to capture it. And third, it's often very periodic loading. It's when it rains, and you get a huge loading of phosphorus in a very large flow, and then when it's dry, nothing. So it's very hard to design technologies to capture phosphorus successfully in this very difficult setting. So maybe we should think of another way of doing it. Don't approach it from the treatment point of view, treatment and capture. Let's try a different point of view to making this P from the drainage and the erosion more sustainable. And that is, let's minimize the loss of the P from the fields in the first place. Let's not lose so much from the fields. And so we want to produce a phosphate-containing fertilizer that is more available to the plants, so we can apply a lot less P, so that a lot less P is going to be discharged when there's a rain event. Okay, so this is the general concept of it. I'm sure everybody in this room has seen a chart something like this. It's just showing the availability of phosphate by soil pH. And you can see in the center part here, whoops, I hit the wrong button there. In the center part here, you've got the sweet spot. This is where the phosphate does not precipitate very much and is available to the plants, but at high pH or low pH, you get a lot of precipitates. And the low pH, you get the iron and aluminum phosphates. The high pH, you get the various calcium phosphates. So it makes it unavailable. You apply a lot more phosphorus than the plants need, and a lot of it runs off. Now, it's not so difficult to fix the low pH problem. You can add lime and to raise the pH, and lime is cheap and easy to find, but it's not so simple for the alkaline soils. And that's what I'm going to focus the rest of my talk on, is what might we do using microbiology or environmental biotechnology to try to improve the situation for the alkaline soils, making the phosphorus more available so we can use less and get less runoff. So how can microbiology help? So the general principle, the overarching general principle, is that we want to prevent the precipitation of the phosphorus, which makes it unavailable. We want to make it more available. and then we don't need so much. So I'm going to talk about two possible microbiology approaches to doing this. The first one is to take advantage of microalgae, who have the great ability to rapidly absorb a lot of bioavailable phosphorus. And then I'm also going to talk about fortifying fertilizers with ammonia oxidizing bacteria to produce acid, so we have in situ neutralization of alkaline soils. So let's do both of them. Number one, using microalgae to absorb phosphate. First of all, what are microalgae? These are photosynthetic microorganisms that are constituted by green algae, that's shown here, that's one Cynodesmus, one green algae, and then our cyanobacteria, which is shown in the picture above. So they're photosynthetic, they use sunlight as their source of energy, they use inorganic carbon, we often call it CO2, as their carbon source, and water is their electron donor. So these are all things that are around. The sun's around, CO2's around, water's around. So we don't have to go out and find anything exotic to grow these organisms. Now, what we're going to take advantage of is these microalgae's ability to scavenge phosphorus at very, very low concentrations. They developed this evolutionarily over many millions of years because they need to. These organisms in their normal environment are exposed to really low phosphate concentrations, unless we put in a lot of extra phosphate because of eutrophication. So they have to be adapted to be able to grab any little phosphorus molecule that comes by and hold it because it's so precious to them. And it's also why, if you look at the standards for protecting water quality from eutrophication, you see that the concentration goals are really low, something like 0.02 milligrams of P per liter. And so these organisms live in these environments, and they're very capable of scavenging very minute concentrations of phosphorus. So how do they do this? Well, they do this by absorbing it or complexing it with amine groups. Amine groups are part of amino acids. Amino acids are part of proteins. So these organisms have proteins on the inside of them, they have proteins on the outside of them. Both of these proteins have amine groups that can complex or bind to phosphate. And that's shown here, simply here. Here's the amine group, there's a phosphate, and they bind to form a complex. And there's a chemical equilibrium representation of it. And they're very good at this. They combine it quite efficiently and rapidly. So how does that work? Well, if you look at this diagram here, what you see here is that we got some inorganic phosphate in the bulk solution. BS stands for bulk solution. That's the water surrounding the organisms. It can rapidly be complex to the extracellular polymeric substances. This is the outside of the cell, or to the internal amine groups here. And so when there's a lot of phosphorus out there, it quickly goes in here or quickly goes here. And then when the organisms need the phosphorus to grow, they simply release it and it comes back and it can go in to spur their growth. So it's naturally bioavailable. So how do we know this is true? So we've been doing a lot of experiments on this topic recently because we're really interested in how phosphorus controls the growth of our microalgae. So here's some results from an experiment in which we exposed some microalgae in the dark, no light, in the dark, so they're not doing photosynthesis. And these were algae that had been grown with different amounts of internal and external phosphorus. And what you can see is the top graph is the uptake, very rapid uptake of phosphate, and these is the buildup of the phosphate complex on the outside and complex on the inside. And particularly if we start with organisms that have low phosphate to start, they can build up a lot very quickly. So within about 2 hours, they've almost saturated their ability. They're taking up a lot of phosphorus. And that amount of phosphorus that they take up is correlated to the protein content, because the protein is where the amine groups are. So this is just correlation here. Whoops. correlation for the extracellular phosphate. Okay, so now the organisms have the phosphorus. They sucked it up really fast, but is it available, and can they use it, and could any organism use it? And the answer is yes. So if you now run that similar kind of experiment, but in the light, it looks like this. So here we have three experiments run in parallel with different amounts of phosphorus. A lot of phosphorus, medium amount, hardly any at all. And the light is on the whole time. So what happens is initially they take up the phosphorus, as you can see here, and they don't really grow much. This is the growth. This is the biomass growing. Not much happens because they're taking up phosphorus. Eventually they've taken up the phosphorus and the organisms start to grow. Now the thing to note here is that the growth rate of these organisms is the same, whether we have a lot of phosphorus still in the water or zero. So the phosphorus in the water isn't controlling their growth. It's the phosphorus that they have absorbed by this complexation reaction. That is what controls it. And that's shown in the next graph. If we just look at the left side, this is for the internal phosphorus here. You can see that this is the rapid uptake here, and then after a while, it's taken up enough and they start to grow and you get this depletion. So first it takes it up, and then it depletes it, and these organisms are growing. So this is naturally bioavailable phosphorus. So how might we do this? Well, we could grow our microalgae, and I'm just showing here some facilities for growing them at our university. We would then, when the organisms are depleted of phosphorus, we would then harvest them. We could then mix them with phosphorus as part of the production of the fertilizer, as you can see down here. And we would then have fertilizer with much more sustainable P, because the P would be bound to the microorganisms, the amine groups, not just free P. And this should greatly reduce the amount of precipitation and loss of availability. Okay, second possibility here of applying some environmental biotechnologies in a novel way, and that is to fortify the fertilizer with nitrifying bacteria to lower the alkalinity and pH. So what we want to do is we want to create a system where the organisms and the conditions that these organisms like are commensurate with what goes on in fertilized soil. And so we need the bacteria and the soil and the plants, all this to be working together and be, have a complementary setting. So nitrifying bacteria use ammonium and they produce nitrate. These ammonium and nitrate, of course, are well-known nitrogen fertilizer components, so that's good. Two, the bacteria are aerobic, the soil is aerobic, so you've got oxygen, it works fine. So far, everything is good. The things the organisms need are compatible with the way the soil system works. So what about these nitrifying bacteria? Who are they? So they come in two groups. First is the ammonia oxidizing bacteria, or AOB, and they take ammonia and they oxidize it using oxygen to produce nitrite. So there's ammonium using oxygen, forming nitrite, and forming acid. So they form a lot of acid. There are lots of known species. I just listed two common names here. They're obligate aerobes. They must have oxygen. Then the nitrite can be further oxidized using more oxygen to nitrate. And again, there are many strains that use this, and I've listed a couple here. They're also aerobic. Now, when I looked at this, I said, well, I'm interested in producing acid to neutralize some of the base in the alkaline soils. It's only the first step that produces the acid. So maybe I only want the first step and I don't want both steps. I don't know about that, but perhaps if plants are capable of using nitrite instead of nitrate, perhaps we could just use one step out of the two. It makes it a little simpler that way. Okay, so, but whether we use one step or we use both steps of the microbiology, we have a situation where we take and we're moving from the right side more to the center side here of this diagram, making the phosphorus more available in the field. So what about nitrifying bacteria? What about them and the conditions that will stimulate these bacteria so they'll really work well in this setting? So I've listed some key things here. I mean, they are obligate aerobic. They have to have oxygen. Fortunately, soil is very aerobic unless we just put in way too much water. So if we keep it from being waterlogged, we'll have oxygen. So that will stimulate the organisms. 2, these are autotrophic microorganisms. Autotrophic means they use inorganic carbon or CO2 as their carbon source. They do not use organic material. They don't need any added organic material. They will not use the soil's natural organic material. In fact, they will add to the soil's organic material. So that's a good thing. 3, they do grow in water. All microorganisms grow in water, and so you do need to have a reasonable moisture content in the soil. Typically, about 10% of the soil moisture holding capacity will be quite good. Now, the last point here is very important. These nitrifying bacteria are very slow growers. They don't grow fast. And so what this means is that to make this strategy work, we will actually have to add the organisms in with the fertilizer, or at least in the fertilizing process, because they're slow growers. They're not going to grow up in a day or so. So we'll need to add a substantial amount of them. So we'll have to grow them and add them into the mix. And it might look something like this. We grow the microorganisms in a big reactor like this, harvest out the cells, and we add the cells with some ammonium and the other constituents to form a fertilizer that has the intrinsic ability to do this in situ reduction of the alkalinity and pH. Now, before I end, just one last thought. As I was researching all of this, I came across a lot of information on testing of bioavailable phosphorus. I'm sure most of you know a lot more about this than I do on this, but I've learned that there are a lot of methods, and I've listed a lot of the common names. I'm sure most of you recognize these names. So there are a lot of options out here, and the key thing is they don't give the same answer. Even when they're used on soils where they're supposed to be optimized, they don't give the same answer. So this is actually distressing, because how do we know if our sustainable P-available fertilizer with our microorganisms in it is actually giving us more bioavailable P? So maybe we don't have adequate methods to assay this already, and that would mean we would perhaps need to develop some more appropriate methodology to assess the bioavailability of phosphorus in this system where we're attempting to make the P more bioavailable through microbiological means. Okay, I'm coming to the end here, and I'm going to just summarize briefly the main take-home messages of my talk. 1, water quality needs are really important. They dictate the need for us to greatly reduce the amount of phosphorus that is being eroded from agricultural fields. It's the #1 cause of eutrophication worldwide, and we've got serious problems worldwide, more than 400 documented cases. For the agricultural applications, perhaps our best strategy is to make the phosphorus a lot more available so that we do not have to add so much. And it will be taken up rapidly by the plants, and we won't have the huge runoff problems that we do now. How would we do that? I've offered 2 possibilities. They make sense from the microbiology point of view. I'm not an expert in agronomy and soil fertility, so those concepts will have to be merged with anything I say, but from the microbiology point of view, these make sense. 1 is to bind the phosphate to microalgae, who have very strong capabilities to rapidly absorb or complex phosphate that is bioavailable. That's how they live, and we can take advantage of this if we design the system correctly. And the last one is the possibility of actually having an in situ production of acid to reduce the pH and the alkalinity of highly alkaline soils, and therefore making the phosphorus in whatever form we apply it less precipitated and more available. So with that, I'm coming to the end here. I do want to say that if anyone would like to chat with me about any of these things, we have two choices. I will be here for the rest of the day. I have to go home tomorrow, so it's only today. However, at the bottom you can see here's my email address, so if you don't catch me today and you have something you'd like to discuss, please send me an email and I'll be happy to respond to you. So with that, thank you very much, and I look forward to having a few questions. Thank you.